Hello, everybody. I'm very pleased to introduce Marco Tulio Angulo for today's talk. Marco Tulio is in Querétaro in Mexico, which is a wonderful place, as I just learned. So he's going to talk today about coexistence holes in ecological systems. So please, Marco Tulio, the stage is yours.
Thank you very much. Thanks everyone for coming. Thanks to the organizers for the opportunity to share our work. So today I will tell you a little bit about how can you apply algebraic topology to understand a fundamental problem in ecology. And it has to do with this word that I am writing here, obstructions. And obstructions will be related to the coexistence of species. So my name is Marco Tulio, and you can contact me using this by email or by Twitter. So how I like to conceive this project is something like a counterfactual. What will happen if Poincaré, that I'm pretty sure that you know very well, he's probably the father of modern topology. What will happen if Poincaré had met this guy? So take a minute to look at him. I guess that you can know that his name is Charles Darwin, the father of modern biology, probably also the father of ecology. And he has a very interesting phrase in his great book, The Origin of Species, in which he describes the mystery of mysteries. And the mystery of mysteries is precisely why do we see so many species in the world? Why is not just one single species that is dominant and there are no other species different from it? So what I will talk today is somehow a combination of these two guys. How can you combine topology from the side of Poincaré with ecology from the side of Darwin to answer a fundamental question? And let me try to introduce you to this question. So to introduce you to this question, I need to tell you a few words about ecology, and in particular about community ecology. So first, what is the meaning of this word community? And for ecologists, a community is simply a collection of species that you observe in some place and at some time. So you can have in mind, for example, if you open the door of your house and you go outside, you have a garden, and the collection of species that are in your garden is a community. The collection of those species can be, for example, grass, some animals, some insects, and so on. So that is a community. And one of the main questions of ecology is why the communities in nature look as they do, as I was just telling you. Why are they so diverse? And the diversity of these communities, it's really striking, and it happens not only in tropical forests like in Mexico, where you can observe thousands of species living on the same place, for example, in a tropical forest, but it also happens inside you. So for your information, in your gut, you have a community of animals, of bacteria, that perform essential functions for your health, and they are very diverse. Approximately, you will have 10,000 different bacteria in your gut, and all of them are required for you to be healthy. So the question is, why communities in nature are so diverse? And one main observation that ecologists have done is that communities not appear spontaneously, but they are assembled in a process in which one species is assembled at a time. And the classical picture to understand that statement is this one. So this is a forest, and what is our idea of how a forest appears? So you start somehow with an empty field. Then the first species arrives. In this case, it can be a type of grass that is called crabgrass. Once that first species is established, then you can allow for new species to arrive. For example, not only crabgrass, but now you can also have some wheat. And then you continue adding species until you get to a point that you will recognize as a forest. And that is called the climax, or at least the ecologists have that name, the climax of the forest. So one main observation here is that there are a lot of constraints. In particular, constraints on which species collections can coexist, and then you can observe, and which species collections cannot coexist, and then you cannot observe. So, for example, if you try to put a tree at the beginning of the assembly, you will see that the tree cannot survive in that environment. It does not have enough partners to survive. And similarly, if you try to introduce grass at the end when the forest is fully assembled, you will see that forest cannot survive there because there is not enough light. So there is this fundamental constraint in which species can you assemble at different stages of the assembly. So let me try to be more clear about this assembly process using a very simple example. So I would like you to imagine this hypothetical ecosystem or community that has three different species of bacteria: the blue bacteria, the yellow bacteria, and the pink bacteria. And then the assembly process that is analogous to this forest that I was describing is the following. So it's the assembly process. So you start with the empty environment. Then the first species arrives. Let's imagine that the first species that arrives is the blue one. If the blue one can survive in the environment by itself, then it will survive or it will coexist. And you will see that the environment now it looks like blue. Let's say. Imagine that this is a forest or some part of the garden outside your house. So initially your garden was just dirt. And then the blue species arrive, and then your garden looks blue. Then a new species can arrive. For example, let's imagine that the pink species arrives. Then if blue and pink can coexist together, then you will see that your garden now looks blue and pink, and you will observe this outcome. And then different species can arrive one at a time. For example, the third species to arrive can be the yellow one. But something that may happen is that now the yellow species, the blue species, and the pink species cannot coexist together. So this community you won't be able to observe. And then the end point is somehow not realized. And you will only be able to observe your garden looking blue and pink. So what I want to emphasize from this example is, again, the constraints. So some species collections coexist, and then you can observe your garden either blue, or you can observe your garden blue and pink. But some other species collections cannot coexist, and for example, this one cannot coexist, and then it's impossible for you to see your garden blue, pink, and yellow. So ecologists actually have been studying this problem of assembly for more than 100 years. It's, in fact, one of the oldest problems in ecology. And they have mostly focused on this, what they call the endpoint. Essentially, what is the maximum number of species that you can pack or that can coexist under given constraints? But the question that we want to answer is a little bit different, because we're interested more in the process. And the question we ask is if there are fundamental yet non-evident obstructions before you reach the endpoint. So somehow in this process of going from the empty environment to the larger community that you can assemble, if you can find in the middle obstructions. But our work tries to answer that question, and I want to list just a few of our main contributions, and then if you are not interested anymore in this talk, you can leave and be happy. So the main contributions of our work is first to introduce this idea of coexistence hypergraphs as a natural mathematical object that can describe the coexistence relations between any number of species. And this point of view is important, or we feel is important, because then the obstructions that I was telling you turns out to be the topological holes that these hypergraphs have. And in particular, I want to tell you that there will be two types or two classes of topological holes. The first type, we will call it assembly hole, and I will be more precise about this in a minute. But for your intuition, an assembly hole is a collection of species that does not coexist, but you can ensemble it from a sub-collection of species that coexist. And perhaps the idea that you can have in your mind is something like a computer. Imagine the computer that you have right now in your desk. It is made from integrated circuits, the parts or the components. And imagine that one scenario in which all the components of your computer work, but then you ensemble them together and the computer does not work. So you have this case in which the components work, but once you ensemble them, the full thing does not work. It's the analogy in ecology is that you have a set of or sub-collections of species. Each of them coexist, but then when you ensemble all of them, the result does not coexist. So that will be the assembly hole. And the second type of obstruction or topological hole, we call it a disassembly hole, and it's kind of the opposite scenario. You have a collection of species that coexist, but that collection cannot be assembled from sub-collections that coexist. In other words, it's something like, imagine that your computer is a thing, an ensemble, and it works. But if you take out one of the parts, let's say one integrated circuit, it doesn't work. So it's something that works, that is made from components that do not work. And that's the idea of a disassembly hole. And it turns out that both of these things can be characterized by as topological properties of the coexistence hypergraphs that I was telling you in the first point. And finally, what we did is to analyze experimental or empirical ecosystems to see if they have these assembly holes or disassembly holes. And what I want to discuss is how these two properties, assembly holes and disassembly holes, are actually overrepresented properties of the ecosystems in nature, and that will have some consequences that I will try to discuss at the end of the presentation. And if you want to know the details of the mathematics or how it relates to existing works, I invite you to please check our work, Coexistence Holes, that was recently published. So now, if you are very bored, you are free to go and use better your time. If not, I want to continue with the following plan. So this will be the outline for the rest of the talk. First, I want to discuss this idea of coexistence hypergraphs that I was telling you, and how can we use coexistence hypergraphs to define this idea of coexistence holes that I was just discussing. And remember that there will be two types of coexistence holes. The first type will be assembly holes, and the second type will be disassembly holes. Then I will discuss how can you uncover or discover these coexistence holes in large ecosystems. It turns out that usually ecosystems will have, I don't know, 20, 100, or 1,000 different species. So then the coexistence hypergraph will need to be embedded in a very large dimensional spaces. And then to identify the holes, we will need to use homology theory. But it turns out that the homology theory for hypergraphs is not as complete or as efficient as for simplicial complexes. So I will tell you a little bit of how you need to adapt the homology theory to work on hypergraphs. Then on the third point, what I will discuss is the application of these theoretical tools based on algebraic topology and hypergraphs to real empirical ecological systems, in particular to microbial systems. And then I will try to suggest some take-home messages. Okay. So perhaps it's a good time to see if there are some questions, as I was asked.
No, at the moment there are no questions, so please continue.
Perfect. So actually, let me start.
I have a question. Sorry to interrupt.
Yes, please.
Just a curiosity. Of course, I'm I'm not familiar with your work, but since you use the word obstruction and holes, etc., I wonder if you will run into multi-valued potentials somehow defined in your hypergraphs.
No, not at all.
Interpreting holes exactly as in topology, so to speak.
So the interpretation that we are using of these topological holes for ecology are obstructions in the assembly process. So somehow you are adding one species at a time. You start with 1, 2, 3, but then you get to a limit. And that limit will be an obstruction that is precisely characterized by the hole.
Oh, I see. Okay. Thank you.
Okay. Thank you for your question. Okay, so let me start with the beginning, first telling you a little bit about coexistence hypergraphs and how you define these coexistence holes. So coexistence hypergraph, let me tell you how they work using an example. So I want to use again this example of 3 species, blue, yellow, pink. And let me imagine one hypothetical community that looks like this. So you will have the following coexistence outcomes. So imagine that it's the grass or the garden of your house, and then it turns out that if you put blue alone, it will survive, and you start with blue, and after 10 days, then your full garden is blue. If you put blue and pink together, they also coexist. And I will encode this coexistent outcome as this node in the hypergraph. I will encode this coexistent outcome as this this edge in the hypergraph. But then let me imagine a case in which the 3 species together don't coexist, and then the interior of this triangle needs to be empty. So what this coexistence hypergraph is telling you is that 1 survives alone, 2 survives alone, 3 survives alone, 1 and 3 can survive, 1 and 2 can coexist, and 2 and 3 can coexist, but the 3 species together cannot coexist. So that will be an example of a coexistence hypergraph. Another example of a coexistence hypergraph that will be useful for us for thinking is the following one. So this one has these coexistence outcomes. So species 1 alone cannot survive. Species 2 alone cannot survive. Species 3 alone cannot survive. Species 1 and 3 cannot survive. 1 and 2 cannot survive. 2 and 3 cannot survive. And the only possibility for the species to coexist is if the 3 of them are put together. Otherwise, they won't be able to survive. So in general, this coexistence hypergraph, to build this coexistence hypergraph, you need to define what is the pool of species, precisely which species can you ensemble. That pool of species will be the vertex set of your hypergraph. And let me suppose that there are, in general, S different species in your garden. Then you need a function that tells you if some collection of species coexist. And if it coexists, it will, this function will return a 1. And if they don't coexist, then this function will return a 0. And then the coexistence hypergraph is simply the collection of all the collection of all the set of all subcollections of species that coexist. So all collection of species such that they coexist, and that will be the coexistence hypergraph. So now let me go to the more interesting part, that is what the hell is an assembly hole. And the archetype that you should have in your head of what is an assembly hole is precisely a typical hole in the hypergraph. So in the previous example that I was discussing, this will be the assembly hole. And what it represents, if you reflect a little bit, is a collection of species that do not coexist. That is, if you put together 1, 2, and 3, that collection of species will not be able to coexist. However, that collection of species could be assembled from a set of subcollections that do coexist. In other words, you can assemble it from the boundary. And that will be the interpretation of what the hell is an assembly hole. So what I just said is written here. And more generally or more abstractly, a coexistence hole will be simply cycles in our coexistence hypergraph that are the boundary of node chain, and it's a kind of the usual definition of what is a topological hole. Perhaps it's not so common because it's not on a simplicial complex, but it's now on a hypergraph. So that is the notion of an assembly hole. Another possibility that is interesting for ecologists is this guy. And this guy will be the archetype of what is a disassembly hole. So a disassembly hole, if you remember this example, is a collection of species that survives together. But if you take one of the part, it won't be able to. So I was telling you about this idea of disassembly holes, and the archetype was this picture. And if you remember, this picture is a collection of species that coexist together. So 1, 2, 3 can coexist together. But if you take one of the parts, let's say if you take 1 and 3, they won't be able to coexist. If you take 2 and 3, 1 and 2, they won't be able to coexist.
Marco Tulio, there's a question.
Yes.
Could you speak up, Joshua, or should I read it?
Okay.
Sure. So let's say you have species 1, 2, and 3, and they cannot coexist. But let's say 1, 2, and 4 can coexist. Is it possible that like 1, 2, 3, 4 coexist,
yes.
even
Yes.
though 1, 2? Okay.
Yes, in principle, any possibility is admissible or feasible. So that example that you are saying, it's totally possible, and it will depend on the particular ecosystem that you have.
Okay, thank you.
Thank you for your question. Okay, so this assembly hole will be species collections that coexist, let's say 1, 2, 3, but they cannot be assembled from covering subcollections that coexist. In other words, they have to be somehow put together at the same time, or otherwise you won't be able to observe them. So one example of this assembly hole is, as I was telling you, this collection 1, 2, 3. And more generally, how to identify them is using the following trick. So you start with the original assembly hypergraph, and what you will do is to calculate what we call the missing boundary. So all the boundaries that are missing from the hyper edges of your original hypergraph, that will give you a new hypergraph that we call the disassembly hypergraph. And if you calculate the holes in that disassembly hypergraph, that will give you the disassembly holes. So in general, the disassembly holes are the topological holes of the disassembly hypergraph constructed by calculating the missing boundary. So in general, what you will find is that coexistence holes, both of assembly and disassembly of different dimensions, can simultaneously exist. And if you have n species, you can imagine, for example, n equal to 100 or 20 or something like that. Then to identify those coexistence holes, you will need to embed the hypergraph in an n-dimensional space, let's say 100-dimensional space. And that is that will make very difficult to grasp just by inspection if there is a hole or not. And just to illustrate this point, you can imagine this example. So this is an example of an assembly hypergraph for a community of 12 species. From inspection, you can identify some assembly hole of different dimension. This one, and these ones are 3 assembly holes of dimension 1. You have two connected components, so you have 2 zero-dimensional assembly holes. And that is more or less easy to grasp just by inspection. Then you can calculate the missing boundary. And you will get your disassembly hypergraph that looks like that. And in that disassembly hypergraph, there are some holes that you can easily grasp just by inspection. For example, this one. So this is one-dimensional disassembly hole. But it will be difficult for you to see that this guy here is actually another disassembly hole of dimension 2. So To solve this problem, somehow what I am trying to suggest is that you need some systematic tool to identify these assembly and disassembly holes in higher dimensions. And that's precisely what get us to the next point of how can you uncover these coexistence holes. And the natural tool to use is homology theory. So just to remind you, if you want to find the coexistence holes in all dimensions of your coexistence hypergraph, what you will need to do is to calculate the homology of these coexistence hypergraphs. But one challenge that is important is that to calculate this homology of hypergraphs, we only have efficient algorithms for a special case in which the hypergraph is a simplicial complex. And in that case, we actually have very efficient algorithms like RIPSER. So to solve this issue of how can you calculate the homology of arbitrary hypergraphs that might not be simplicial complexes, what we suggest or what we introduce is the following method. So this is an efficient way to calculate the homology not only of simplicial complexes, but of general hypergraphs. And the trick is to transform the hypergraph into a simplicial complex, actually. And let me illustrate the trick using one example. So let's imagine that this is your coexistence hypergraph, this guy. So you have 4 species that coexist like this. And what we will do is to actually subdivide it, very similarly to how you apply the barycentric subdivision to simplicial complexes. And when you subdivide it, what we will build is this subdivision. So please notice that in the original coexistence hypergraph, you don't have this edge between species 2 and species 3. That is, species 2 and 3 together, they don't coexist. So it is clear that this guy here, it's a hole. So when we apply the subdivision, well, what we do is to assign to each hyperedge one node. And for example, the node corresponding to this hyperedge is this one. The node corresponding to this hyperedge is this one, and so on. And then you connect them by inclusion. And this, from this subdivision, I hope that you can easily see that it's evident that now you have a hole between species 2 and species 3. And importantly, the subdivision, note that it is a simplicial complex. And what we can actually prove is the following theorem, that if you denote by S of H the first subdivision of the hypergraph H, then we can prove that the homology of the subdivision is identical to the homology of the original hypergraph H. And somehow the proof is not trivial for me. For example, in the theorem is claiming that just using one subdivision is always enough to somehow preserve the homology of the original hypergraph. And it's tricky to show you that it's always enough to have one subdivision, that is not necessary to do 3 or 4 subdivisions. And the importance of the utility of this. Yes.
In terms of commuting the homology of H is not a simplicial complex.
Correct.
So how is the homology of H computed? S of H is a simplicial complex. So you've got boundary maps, you've got boundaries. But in this thing, H itself, you may not have, the vertices may not be existing. How do you compute the homology of H?
Thank you for your question. Actually, I will let Luis, that is my mathematician, to answer that question. So please, Luis. Okay, then perhaps.
Sorry. Can you hear me?
Yes, we can.
Yeah, what we do is we understand the hypergraph H not as a hypergraph, but as a topological space. And it's the following topological space. You put a vertex in the you put the interior of the cells. In other words, if you have this, the 1, 2 cell, you put the interior of 1, 2, the interior of 1, the interior of 2, and then you get the interior of 1, 2, 3, and so on. In other words, take for the topological space, which corresponding to a hypergraph, take the interior of all the hyper edges, and in that way you get a topological space, which is not a simplicial complex. It's just a topological space, but it has a homotopy type of this simplicial complex.
Okay, good. Thank you.
Thank you, Luis. Okay, so one important remark, or what is useful from this theorem, is the fact that now S of H is a simplicial complex, and then we can apply, for example, Ripser to very efficiently calculate the homology of the original hypergraph H, just by computing what is the homology of S of H. So this is useful because now we have an efficient tool to calculate the Betti numbers of a hypergraph H for an arbitrary hypergraph H. And I just remind you that the Betti numbers, it's a vector in principle of n different numbers, where this number beta k is the number of k-dimensional holes that your hypergraph has. So if you calculate the Betti numbers of the assembly hypergraph, what you will calculate is the assembly holes. And if you calculate the Betti numbers of the disassembly hypergraph, then what you will calculate is disassembly holes. So to avoid you of any troubles, what we did for you is to build to pack all these tools together into a software written in Julia that we call Coexistence holes, and that you can easily download and use in your computer. And in fact, we have tutorials if you want to use it either using Julia or R. So you can very easily calculate your favorite coexistence holes. Okay, so with this, what I have discussed is somehow the tools that we introduced. So actually, what I have discussed is first the concept of assembly hole, disassembly holes, and then the tools to discover disassembly and disassembly holes in hypergraphs. Now, what I want to discuss is how you apply these tools to actual empirical ecological systems. And what we did is to use these 5 different experimental microbial systems that were studied before by different authors, by Vandermeer, Friedman, Stein, Venturelli, and Design, and that they had different number of species. So the smallest number of species is, for example, 4 in the work by Vandermeer, and the largest system has 14 different species. And actually, they describe different ecosystems from purely academic or in vitro experiments, like the work of Vandermeer, to more realistic systems, for example, in the work of Venturelli, that these systems are related to the gut bacteria that humans have, or that healthy humans have at least. And the collection of experimental systems that we can use for this methodology is actually very small, because we need 2 properties to be able to construct the coexistence hypergraphs. First is that we need high resolution temporal data of your species. You need to know what abundance of the different collection of species are there. And so also something that was important for us is that the mathematical models that we can leverage on to build the coexistence hypergraphs were previously validated, so we don't need to worry about the validity of the models. Okay, so with this, let me show you a first glimpse of how they look. So first, I want to discuss how the coexistence, how the assembly hypergraph looks, and how the coexistence holes are. So here I'm showing you only 4 of those 5 experimental systems, from Vandermeer with 4 species in the left, to Venturelli with 12 species to the right. So these visualizations are even interesting by themselves. For example, something that you can easily grasp from Vandermeer is that the coexistence, the assembly hypergraph is actually a simplicial complex, right? It actually looks like a lollipop. Then in this case, what you see is that it's obviously only one connected component, and it does not have any hole of dimension larger than 1. And something interesting that can be is also the number of hyperedges that you observe and the largest collection of species that can coexist. Then this is, for example, what happens with Friedman. This is a system with 8 species. Again, something interesting that happens in this case is that the coexistence hypergraph is actually a simplicial complex. And it looks like a boat, somehow a falling boat. And what you observe, again, is that it has 1 connected component and 2 one-dimensional holes, and you observe some type of number of hyperedge and so on. And something that is that you can observe from these 4 illustrative examples is that as you increase the number of species, what you tend to observe is a higher number of coexistence holes and also of larger dimension. So, for example, if you go to Venturelli, then you cannot read much from the visualization, but now you have 9 two-dimensional holes. In Stein, you have actually 2, if I remember correctly, 4-dimensional holes or 5-dimensional holes or something like that. Okay, so this is what you observe from nature. Then the interesting question is the following: are these numbers just can these numbers be explained just by chance, or are there some ecological mechanism that is setting this number of coexistence holes? So to answer that question, what you need to do is to compare what is the observation that you are making, the number of holes of the real ecological system, with respect to what you expect from just a random ecosystem with the, let me say, with the same properties. And this is what I am trying to show you in these panels of the bottom. So what you will see in these panels in the bottom is blue points that correspond to the actual real ecosystem. And then you will see some gray bars that represent what is expected just by chance, and some gray points that represent what is the expected number of holes of each dimension just by chance, all right? And what you observe is that for small systems, for example, for Van der Meer, you observe that the blue point is below the gray point. And what it means is somehow that in this case, the system is depleted of coexistence holes. In other words, it has a smaller number of coexistence holes than what is expected by chance. But as you move to more complex systems, what you start to observe is these huge differences. Right? And these huge differences represent that at certain dimensions, the number of holes that we are observing are actually 6 standard deviations higher than what is expected by chance, like in this case and this case.
Could you comment on how you build such a graph from data? So, for example, I see in that one, there's 1 or 4, 3 is an edge, but did they like take a gut and just put, bacteria 4 and 3 and check if they coexist?
Let me say yes. So let me say that you do an experiment. You have a jar and you put 1, 4, and 3. Let's say this one in a jar. You put the 3 of them inside. You wait, let's say 48 hours, and you check if they still are there after 48 hours. In this case, you will observe that some either 1, 4, or 3 died, and then they are not able to coexist.
So for this Venturelli experiment, they did 2 to the 12.
No.
Did they check every subset of those 12 species that those coexist?
No, they only did a subset of those experiments. And from that subset of experiments, what we did is to build this mathematical model that I was telling you in the previous slide.
Yeah, I understand. Okay.
And then we use the mathematical model to somehow extrapolate the species collections that we were not able to experimentally do. Is it clear?
Okay, I think so, yeah.
Okay, thank you for your question. Okay, so in this is what happens for coexistence holes, and now let's see what happens for disassembly holes. So something that you observe, for example, for the small systems, remember that Van der Meer is the smallest system. It has only 4 species. Its assembly hypergraph was actually a simplicial complex. And then when you compute the missing boundary of a simplicial complex, it's just the empty hypergraph. So actually, these points should be somehow empty, but if I draw them empty, you won't be able to see it. So what you observe in this case is that you have 4 different components. So one property that you have is that if the disassembly hypergraph has S connected components, where S is the number of species, then that means that the original assembly hypergraph was a simplicial complex. The same happens for Friedman, and then something more complex occurs for Stein and Venturelli, where you observe actually, for example, a huge number of disassembly holes of dimension 2. So again, this is somehow what you observe from nature, and then again, the question is this what we can expect just by chance, or is there some mechanism that is either enriching or depleting the number of disassembly holes? So you do the same trick that I just mentioned. You compare the actual measurement with respect to randomizations. So remember, the gray points are randomizations. The color points, the pink points, are the actual measurements. And what you observe again is some dimensions where the number of holes, for example, in this case, the 64 disassembly holes of dimension 2 are very, very unlikely to be observed by chance. The probability is just 0.00011. So very unlikely, and this means that these are actually enriched by the ecological dynamics. Okay, so now let me try to finish and suggest some take-home messages. So first, let me summarize what the hell are these coexistence holes. So what I suggest is that coexistence holes represent fundamental obstructions either to assemble when you think of assembly holes, or to disassemble when you think of disassembly holes. And probably relevant for this audience is that these obstructions are revealed using algebraic topology. And something relevant is the fact that algebraic topology is actually a very underused tool in ecology. And ecology is actually among the biological sciences. Ecology is probably the science that is more mathematically sophisticated. So this work that I am presenting is probably the first application of algebraic topology to ecology, at least to the best of our knowledge. And what I show you by analyzing experimental systems is that the number of coexistence holes, either of assembly or disassembly, are overrepresented properties of the natural ecological systems that we studied. And the implication of this is that actually the biodiversity in nature is a discontinuous process that is driven by constraint of design. So biodiversity is not something smooth, as perhaps ecologists have imagined for many years, but it has these discontinuities or obstructions that are precisely characterized by these assembly holes. So to finish, let me give credit to the ones that deserve them. So this is joint work with Aaron Kelly, a student from UNAM, Luis Montejano, that also participated with us a few minutes ago, in collaboration with ecologists. So as you may know, these guys are mathematicians, and this was a collaboration with ecologists. So to ecologists, Julian Song from University of Toronto and Sergei Saavedra from the MIT. And with this, I finish, and I want to thank you for your attention, and I will be very happy to try to answer any question that you may have. Thank you.
Thank you very much for this really interesting talk and an application of topology to some field which was totally new to me. So do we have any questions?
Yes, I've got a question. How do you
Yes.
compute the expected number of, say, the expected Betty numbers? Do you
Okay.
somehow generate random complexes and compute their homology, or?
No, what I generate is random ecological dynamics. So remember that to build these guys, for example, this guy, we will mix experimental data with a dynamical model that predicts coexistence of a species. And that dynamical model has some parameters, some ecological parameters. So what I do is to randomize those ecological parameters to generate new hypergraphs that are just what is expected by randomizing the interactions. For example, if in the model species 1 interacts with species 2, then in the randomization, perhaps species 1 now interacts with species 5, but with the same strength. Did I explain something?
Okay, yes, thank you.
So randomization is on the dynamics, not on the simplicial, not in the hypergraph.
Gotcha. Thank you.
Thank you for your question.
Do we have any more questions?
Yeah, so I have many, but so I guess one of them is, can you discuss like maybe robustness to experimental error, right? I mean, if what I mean by that is, if you accidentally added an extra edge in one of your simplicial complexes, this will not necessarily, if you added an extra edge to the Venturelli graph, right, you're not necessarily going to get, Betty number now is like 45, right? So have you explored that? Like maybe if you assign edge weights or probabilities to edge weights?
That's a very good question. We have not, but it's something that we are planning to do, to somehow to study the robustness of the method against measurement errors. And measurement errors are very drastic in ecology, so it's a very relevant question, but we have not explored it yet.
And my second question is I think you guys are I think one generalization, and I think it's a fair one you've been making, is that like kind of species coexistence is static. Basically if you have, okay, maybe I'll turn on my camera and I'll say hi. Maybe my apartment's messy. Essentially, like you're thinking, if you put like 3 different bacteria in a petri dish, when you look after, the populations at some point stabilize, so you can say, oh, these all coexist. But oftentimes, like species coexistence is dynamical. Like even just mathematically, we know like Lotka Volterra, if you observe, if you pick a certain slide, maybe the wolf is very high, but the lambs are dead. So I guess, is there like maybe a dynamical way of viewing how maybe these complexes evolve?
Yes, actually, there are definitions of something that ecologists call persistence. The persistence of a species collection is the fact that no species goes to zero in finite time, let's say. And then these hypergraphs are constructed using that definition of persistence or permanence. And there are efficient criteria to check if a species collection will persist or will be permanent according to some model. But in general, the question is very tricky, and one ends up needing to do simulations to check if some bacteria actually dies. Because perhaps also related to your question, in these hypergraphs, some of the collection of species will not just tend to equilibrium and then stay there, but they will oscillate, or they can have even chaotic motions. And in all those cases, what we assume is that, what we conclude is that the collection coexists, and then we draw the edge, or we place the hyper edge. Don't know if that answered your question.
Thank you.
Thank you for your question.
Any more questions, please? So I have a question. Since you manage persistence, I was wondering, in the beginning you showed this example of the forest growing from one species and then you add more and more species to it. And maybe in this process, along the way, some of the species that you have introduced in the earlier stages later on die, and these constructions that you're making change along the way. So a way to encode that in mathematics would be using persistent homology instead of just homology of the resulting community in the end. Have you thought about doing that?
Not really. Actually, how I conceive this example that you were saying of the forest is something like moving on top of these hypergraphs. So let's say that you start with a species 1, and then you add a species 4, and then you go to this hyperidge. And you then add a species 2, and then you go to this hyperidge. So that is one kind of trajectory of assembly. But you can also go in different directions. Let's say you start with 1, then you add 4, and you go here, and then you attempt to add 3. The species 3 is in the bottom left corner. And since you don't have that hyperidge, then you may collapse, and you either go, for example, either to 1 or again to 1, 4.
Mm-hmm.
And I don't know how you think about this process that you were saying using this persistent homology.
So persistent homology is a process. If you have a simplicial complex which is growing, you're adding things to it all the time or changing in some way, then you're along the way following how the homology changes. And then you have different ways of presenting that. You can use persistence graphs or persistence diagrams and so on. Yeah. And these are really also some kind of descriptors that tell you something about the community. Maybe, I'm just guessing, maybe that would tell you something about the community that you're looking at.
Mm-hmm. Yes, so something perhaps to note is that in this case, the hypergraph is fixed, or the simplicial complex is fixed, because either the species coexist or not. So it's not that you are adding hyperedges or complexes. So they are fixed because either a species coexist or don't coexist. Some other project that we are actually working on right now is precisely the one that you mentioned, in which you have somehow one parameter that, let's say, it's temperature, and then as you change temperature, then it may happen that the hypergraph changes. Right? So perhaps at the beginning it was filled, and then you change the temperature a little bit, and now you find a hole, and then perhaps it breaks into pieces and so on. And then this idea of persistent homology is perhaps the natural tool to address this. But at this point, kind of conditions are fixed. Let's say that temperature is fixed, the pH is fixed, humidity is fixed, and then you have only one hypergraph. And then the questions are about that fixed hypergraph. And something very interesting, as you said, is okay, how the topology of these coexistence hypergraphs change as you change some parameter. And that is kind of a bifurcation diagram, but not looking. Bifurcation diagrams are kind of the basic tool that ecologists use to analyze their ecosystems. But usually they focus on species abundance. So they have this graph of, for example, temperature versus abundance of the species. And then if the temperature is very high, then the species cannot survive, and you observe a collapse of abundance. Something different is in one axis you put a parameter, let's say again temperature, but the other axis is something related to topology, right? Kind of the topology of the assembly and the disassembly. And then you have bifurcation diagrams, not about abundance, but about the assembly and disassembly process that you can use to build that community. And that is something that we are actually exploring.
Thank you. Thank you very much.
Thank you for your question.
So you're, just so I understand what you were saying, you're saying like if you take temperature over time and you just build the hypergraph for each one, maybe after at 50 degrees Celsius, now all of a sudden you're plotting Betty 1 at each time. Now Betty 1 at like 50 degrees Celsius becomes like 5,000 when originally it was 1, something like that.
Yeah.
And that would be bifurcation.
Yeah, and that will, it's kind of a bifurcation plot.
But I think maybe if I'm trying to understand what Neza was saying, I think it's like if you, maybe like in this one on the far left, when you added species 3, the species interaction between 1 and 2 died. But then when you add, let's say, species 5 now in, then 1 and 2 start coexisting again. Does that make sense? Is that even possible biologically? And And I think that would kind of be a motivation. If that does happen, that would be a motivation, I think, for like
something that is useful is to assume that the pool of species is fixed. So, for example, in Vandermeer, if you have 4 species, you only have 4 species, right? So this idea of adding new species is not the best idea, I feel. So, and just imagine that you have 4 species, and what can happen, as you said, is that you start, for example, with species 1 and 2, so and they coexist. So you have that hyperage, and then you try to add 4. For example, 4 is this way. No, and since you have that hyperage, you know that you can move in the interior, and then somehow you are there, right? But then if you want to add now species 3 is here, then you won't be able to do it. Do you see why? Because you don't have a hyperage that covers 1, 2, 3, 4. So they won't be able to coexist together. So you will either stay at 1, 2, 3, or go to somewhere else in this coexistence hypergraph, or to 0.
I'm sorry, I have so many questions. Do you have a dynamic model that would maybe do this? So, for example, the dynamics of just one species, it will just be like a fixed point. Species 2, now you add in the second species, then you'll have, a coexisting fixed point of like 1 and 2. Now you add 3. Okay, now 1 and 2 die, but like 3. Do you know?
Something very useful in this project is the fact that we have like 100 years of theory of how to build models for ecological systems. So, for example, in this case, we are using the very classic lockable Terra system that has a minimum number of parameters that we can infer or adjust using the very limited experimental data that we have. So in that sense, we can make all those experiments that you suggest. Something that we have been thinking, in fact, is that we need to add somehow a direction to these edges, right? In the example that you were saying, let's imagine that you try to ensemble 1, 4, and 3. So we know from this hypergraph that they won't be able to coexist. But something that will be very useful is to also encode what is the result of that assembly. For example, it may happen that 1, 4, and 3 will result in 1, 4, for example, right? So we need to add that direction. And in that case, what you obtain is actually a list of what is kind of the initial community, initial, and to final. And you have a list like this. So, for example, you start with 1, 4, and 3, and perhaps you go to 1, 4. Then you go to 1, 2, 3, and perhaps you go to 1 only, and so on, right? So it becomes just like a list of outcomes of the coexistence outcomes that I just draw in the first slides. Did I say something?
No, yeah, I think maybe just so I can process that. You're saying, like, for example, if you had 4-dimensional generalized local Terra, you put in an initial condition, let's say 0.1/3, 1/3, 1/3, 0, and then now will this now converge to a solution that's only supported, let's say it now goes to the fixed point 1, 0, 0, 0, something like that, right? I see. That's really interesting.
But that is something that we are also considering, but we are not sure of the tools we need in that case, because. But yes, that might be something useful also for ecologists.
I've tried something before like that. Thank you.
Thank you for your question.
Marco, from what you said, you mentioned the effects, the influence of a physical parameter on your changes of topology. It's a nice puts a light on a nice connection between, if you like, phase transitions on the system and topological features. So that's quite interesting to establish a bridging language between phase transitions in dynamical systems, just more generally now, and the topology of your hypergraphs. So that's an interesting thing.
Yes, we are
Thank
very
you.
excited about that, and in particular because these phase transitions or bifurcation diagrams is a language that ecologists use. But for us, it's very important to use their same language. We don't want to use. We want to use as much as possible their language, and bifurcations is something that they love to use. Thank you for your observation.
Thank you. I was on mute. So if there are no more questions, I would like to thank you again warmly, because this was really very, very interesting talk. And thank you for all the questions and your very informative answers.
Thank you everyone for coming and for the invitation. I hope to see you soon.
So bye-bye.
Fico has a question, I think.
Oh, there's one more question.
Maybe it's a question for Luis Montejano. You say to every hypergraph you associate a topological space. And let me repeat what I understood of your answer. If you have a hypergraph, you can associate a simplicial complex maybe in a different way, which is simply add to the hypergraph all subsets of members of the hypergraph. Then you do have a simplicial complex. And if you have a simplicial complex, you obviously have a polyhedron or a topological space associated to it. Now, the space associated to the hypergraph can be obtained from this simplicial complex, or by removing some simplices. This correct? Or maybe I don't know if I can ask you, abusing your time, to explain again how you construct the topological space from a hypergraph.
Put your vertices in the vertices of a big simplicial complex. And every time that you have an edge of the hypergraph, take the interior of this simplicial complex and take the union of the interior for edges of the hypergraph. And then this topological space is what intuitively the ecologists had in mind to understand the hosts.
Okay, yes.
And then what we simply do is from this topological space that is the topological space associated to a hypergraph, we construct a simplicial complex by this classical idea of barycentric subdivision.
I think I understand. Thank you.
I apologize. I didn't notice that you were asking a question, so sorry. Any more questions, please? Okay, it seems that there are really no more questions now. And in that case, thank you very much for your very nice talk.
Thank you again, everyone, and for the opportunity to share our work. I hope to see you soon.
Thank you, Luis, and thank you, Marco.